Welcome to lesson 2.3, the last lesson in unit 2 on soil ecology and the soil food web. And we'll discuss in this lesson some of the strategies in conservation agriculture in terms of how avoiding certain practices like tillage and fertilizer use and chemical usage and stubble retention actually allows soil ecology to recover and actually supports the soil food web organisms. And then we'll also talk about how we can cultivate microbes of the soil food web, particularly the microscopic creatures that we want to encourage as the basis of re-establishing ecological processes of the soil in order to restore soils.
So how do we then manage this system? In the sense of how does our management impact this system? So, of course, tillage disrupts the aggregates, the structure, compromises microbial functions, nutrient supply is compromised, and this protection from what we call pathogens is non-existent when we disrupt and we compact the soil. But in conservation agriculture, we can actually increase soil organic matter because we're leaving the particulate soil organic matter pool by the residue that's left on the soil. And up to 35% of total carbon is in that top 5 centimeters of topsoil. So 50 to 75% of microbial activity actually occurs in that 5 top centimeters. So we want to leave that intact. We want to reduce tillage, and that increases microbial distribution in the profile. In Australia, a lot of our crops rely on being fed by rain. Microbial activity is only short-lived, and it's infrequent because it only relies on that rain. So surface soils have short wet versus long dry periods. And so this really reduces microbial activity and therefore food for our plants. But if we actually use conservation agriculture, which means no-till, I would say, but reduced tillage still disrupts things. So no tillage, stubble retention, constant crop cover. If we can increase that, we can increase soil moisture and we can increase microbial functioning. So it's particularly important, this conservation agriculture in rain-fed cropping systems. So really what it does is, once again, residues, aggregates are formed, that habitat for the bigger organisms like earthworms to work in and the fungal networks to function in and those biopores to be formed. And carbon sequestration then protects organic matter and the microbial processes that occur around that organic matter. And diverse micropopulations in varied conditions, example, non-symbiotic nitrogen-fixing bacteria. So these guys actually require reduced oxygen pressure in the soil for their enzyme to actually fix nitrogen. So when we have the various microbe populations, they need different habitats in that. So if we disrupt and homogenize the soil by tilling the soil, of course, none of this can happen. So we're reducing the very beautiful systems, the ecosystem services of particular microbes that cannot function. So we need to conserve them. So water and oxygen entries are obviously added in as well if we have conservation agriculture. It is critical in water repellent soils because 10 million hectares, over 10 million hectares in southwest Australia agricultural soils are actually quite repellent. And you can imagine why, because when droughts happen for long periods of time and the soil is dry and there's no organisms present and organic matter is depleted, water is literally repelled. Water is literally running off. So we need to increase these systems, water entry, water absorption, oxygen absorption for all of the system to work, and so it doesn't any longer repel water. This also increases drainage and water holding capacity. So that's why it's so important in these systems, and decreases water logging and stress and wind erosion. It protects soil and microbes from heat and evaporation. So non-symbiotic nitrogen fixation is one great example of what conservation agriculture can provide. It's the free-living bacteria that don't rely on having a nodule on the plant. So it's not in just leguminous plants. It happens around any plant really, as long as the organisms are fed and present. They use decomposed residues, organic matter that is 50 to 70% cellulose or hemicellulose. And this predominantly happens in termite mounds and around plant roots. So they need carbon energy to transform other nutrients and need cellulitic microbes to access that carbon. nitrogen fixation is proportional to residue and decomposition rates of that organic matter being present in the organisms that can provide that carbon for those nitrogen fixers. And in macro aggregates, there's reduced oxygen tension, so nitrogenase enzyme can actually fix the nitrogen. And we'll go into nitrogen fixation into more detail, but I wanted to demonstrate here how nitrogen fixation occurs naturally and increases the economic value of the crop and increases environmental sustainability if the soil food web is actually present and the organisms are functioning in unison to create very structured soil. Nitrogen mineralization rapidly increases with complete soil food web organisms being present where the whole system functions when a structure is present. And termites and arthropods that are allowed to stay in the soil as a result of not removing them through tillage fix up to 10 kilograms of nitrogen per hectare per year. That's free delivery of nitrogen to your plants without having to fertilize. And that is dependent very much on the residue quality and quantity. And disturbance reduces this aggregation, so we want to minimize disturbance at all cost. It also reduces soil carbon and pore networks. So it reduces the decomposer and nitrogen-fixing bacterial interactions. So we need both bacterial forms. The nitrogen fixers rely on the carbon decomposers. And so therefore we want to maintain that structure of the soil in order for the system to function and bring us gains in terms of food production. And this is a very simplified nitrogen fixation process. And we'll go into much more detail, but obviously from the atmosphere, nitrogen-fixing bacteria such as these non-symbiotic nitrogen fixers make ammonia nitrifying bacteria, make it into nitrate, and then denitrification is when we actually have an anaerobic system and all of that nitrogen that's been fixed by these cute little creatures that really do an amazing job that nothing else on earth can do, denitrify that and put it back into the atmosphere. So we want to keep our nitrogen in the soil once it's fixed. And the problem is that nitrogen fertilizers, just at 25 kilograms of nitrogen per hectare, synthetic nitrogen, actually reduce, obviously, this beautiful system, the free workers, the non-symbiotic nitrogen fixers. So this is why conservation agriculture is so important to maintain this system. The same happens with arbuscular mycorrhizal fungi. Basically, these guys, we need them desperately because they feed phosphorus, nitrogen, and zinc to 80% of plant families, and they provide plant immunity and plant beneficial microbes. They require living roots for survival, and so basically these very essential organisms that allowed plants to come to land eons ago, and actually the two are non-separable basically. A plant cannot be looked at upon as an entity without arbuscular mycorrhizal fungi present, because that's how they're always fed through these fungal networks on their roots. That's how plants have always received food. But they are actually reduced in their abundance when there's no plant cover, obviously because they require living roots just to feed them. And also when there's absence of a host crop. So you might plant a crop that relies on these organisms, but then the next crop you're planting doesn't have them or it's got a different species. So then you're losing that first species that was relying on your first crop. So you want to be able to manage that and understand which of these arbuscular mycorrhizal fungi your crop specifically requires and is symbiotic with. So poor colonization of crop leads to deficiencies. So when there's not enough fungi colonizing your crop roots, it means long fallow disorder. What that means is basically you've just killed off your mycorrhizal fungi or arbuscular mycorrhizal fungi, and we call it a long fallow disorder. So it's something obviously farmers see, and the reason for that disorder is because their crops are not colonized by the very organisms that need to feed their crops. So North Australian grain region experiences long periods of fallow and burning and ploughing, and to control weeds, they even cultivate the soil to prevent the weed proliferation after rain. So this obviously destroys the mycorrhizal fungi, because these arbuscular mycorrhizal fungi have networks. They run throughout the soil profile. They run across and they communicating plant to plant. They associated with every plant. So if you till the soil, you're disrupting that whole system. They're so fragile and they don't just grow back very readily. They need to have continuous structure in the soil to maintain their bodies, maintain their harmful networks. So we can learn crop mycorrhizal dependency to build up the spores that these guys produce, the avascular mycorrhizal fungal spores. And then when you build up the spores and you put your next crop in that is also symbiotic with these particular species, they grow immediately. Everything just functions. There's continuation. So we need to learn this as growers. And this is a beautiful image of an avascule. What that is aba means tree, skule means tiny. So tiny little tree, this avascule is inside a plant cell between the cell wall and the cell membrane. And it literally has its own little nook where the plant invited it in through biochemical communication. And the spore is able to germinate and penetrate through the cell wall into the plant. And this is where the nutrient exchange occurs. This is where the plant delivers its carbon sugars, liquid sugar, into the fungi to feed the rest of the fungi growing out of the plant cell. And this is where it pumps in the nitrogen, the phosphorus, the zinc, every mineral that it gains from the surrounding soil and mines these nutrients for the plant. So that's just to show you the intimacy of this relationship. And of course, there's disease suppression as well. So basically, in conservation agriculture, we show that disease suppression is very real. And that's because, once again, all the beneficial organisms are present, everything's functioning, nothing is disturbed, nothing is under pressure. So there's reduced incidence and severity of disease, even when pathogens are present, and so is the host. And so are the perfect condition for that, what we call pathogen, to be actually active and eating a crop. So these soils, or these conservation agriculture-based soils, are in effect inhospitable to pathogens. And there's general inhibition of these pathogens by diversity of microbe-fauna interactions. So how the little organisms and their predators or their grazers interact. So basically, in that case, the plant is healthy, it's getting all the nutrients. And of course, the habitat is great. So why would there be any pathogens where there's oxygen and the right amount of moisture present and it's not waterlogged? There's also specific suppression though as well, which is specific antagonism. So example of that is when a Pseudomonas bacterium suppresses the take-all of wheat. So this is take-all of wheat, and this is what it looks like under the microscope, which we call disease. But basically, that's what happens when the plant is unhappy. It's literally attracting these organisms to feed on it because it's dying. It needs to be decomposed. But when these organisms are present, and I suggest that there's many more, and there's a lot of interaction happen for this suppression to occur, but it's just the natural immunity that these beneficial microbes provide, then this thing we call disease doesn't occur. So it's just a symptom of unhealthy soil. That's all it is, really. There's no microbial diversity to prevent it from happening. So you can say that there is no immunity in the plant. Population, activity, and composition of microbial community is also very much, therefore, the giver of this immunity to the plant. So basically, disease suppression is a function of that population being healthy. And the conservation agriculture farm management, the practices that are used are key. For example, in studies, it's been shown that 7 years accumulation of carbon, because obviously it's been depleted for so many years. Now when you put back the carbon into the system and the microbial turnover, so how that carbon is being utilized and fed upon, changes microbial communities, and it further, with time and with carbon accumulation, increases suppression. So that was just to show you the different benefits we gain when we conserve rather than destroy soil.
So to summarize and move on to the practical component of it. So we want plant diversity and the different root architectures and exudates to increase the soil ecosystem diversity, to increase our biomass of the different organisms and increase the different groups and the different species to get that biodiversity really enriched. We want to increase our soil organic matter through plant residues as a microbial food source. And so we want that plant diversity and the fact that we're leaving these plants to do their job and to also die their natural death post-harvest. So the soil food web basically are complex interactions, mineralizing plant nutrients. So we want to understand that each trophic level, each feeding level does something different and is essential. And without one being present on the bottom, like if bacteria and fungi were not present, obviously nothing else can feed. And if there are fungi and bacteria but nothing can feed on them, the nutrients are not released. So we've got to just keep harnessing and understanding the idea that we need more of everything and we want to establish everything from scratch. This is why we're going to go into the cultivation of the microbial ecosystem in a second. So decrease soil disturbance and encourage soil food web development as a result to increase that complexity, and allow for biopores and aggregates to be formed by leaving the soil intact without disturbance. So there will be a stable coexistence of microbially diverse groups with cooperative functions in nutrient cycling and plant immunity and plant nutrition. Soil organic matter and soil food webs basically restore the soil structure and function. They speed up above ground succession, they increase crop yields, and they definitely increase carbon sequestration. And we can cultivate and transplant the soil food web microbes and replenish the depleted soils through the following. So I wanted to show you guys and introduce this willy worm flow, I call it, worm farm, which is basically a willy bin, as you can see. And it's the simplest way to create one of these because you can just transform an upcycled willy bin. And sometimes certain councils will give away those bins. So I would just contact the council and ask directly there. You get a wheelie bin. You create 60 centimeter depth from the top of the bin to the bottom where the slats are. So we've got 60 centimeters here, and there's these tubes which are conduit. It's better to get electrical conduit because it's firmer and better to deal with. You can also find this, oftentimes people are just giving it away or buy some from Bunnings, for example. But every 6.5 centimeters allows across this bin for 7 of these to be fed through. The idea is that you're going to harvest your castings from the bottom. Through continuous feeding of your worms, you will end up with nice fungal biomass on the bottom here over time, within a year's time. So that bottom 20 centimeters will be harvestable, and up here in the top 20, your worms will be living in that layer there. And you can open this up and then get a little rake and scrape out the castings from the bottom. So you're never disturbing the top. You're not disturbing the ecosystem. You're just harvesting from the bottom and then using that in your soil. So you can do this also instead of a bin. Another way to do it is in these barrels as well. As long as you've got that 60 centimeters and you've got enough surface area that you feed the worms. And that's just to show you like the different tools you can use to do this. This is an angle grinder. I've now become accustomed to using a jigsaw instead, which is much safer. And you basically just put these bars in again, and you want it nice, and you just want to make some stability here. This is cut way too far, because remember, you want this bin to be stable and not topple over when it's really heavy here. And you can also make a little door for that as well. But basically, to set up the worm farm, you want 20 centimeters of bedding at the beginning here, and you want cardboard, lots of cardboard, maybe 5 to 10 even centimeters worth of cardboard. Because remember, you want it not to flow out to start with and just fall to your harvesting area. You want to develop that biomass over time here nicely. And you want to use shredded paper and coir after that, something that's not going to decompose easily. And you might want to add a little bit of good compost to inoculate the worms, to give them some good microbial diversity to start with. You want to soak and squeeze the matter with 60% moisture. So you want some drops to fall out, and you just want to make that bedding really nice and comfy for them. All kinds of organic matter that you can use. Probably shredded cardboard is better than just paper, because cardboard won't just stick too much to itself. But diversity is always key. So you want to make that first layer very diverse for them as well. Fluff it out, add some soil. For grit, you can do that if you want. That's not necessary, but people say that it helps. And you want to add some red wigglers, Yersinia foetida, 1,000 worms or so, which is half a kilo, and add a thin layer of food and carbon material. And when the food is gone, you add some more. So this is just a simplified version of looking at this, but you really want some carbon as well on top of the food layer. So you want to feed everything except for like citrus, large amounts of onion or anything like that. You just want to make it nice and simple for them to digest. So soft foods, of course, no dairy or meat, because that will attract all kinds of other organisms. Weeds will be okay if you've got some. You don't want to ever overload and overfeed them. You want to have the food disappear within 3 days, and if it hasn't, don't feed them again, and work out a system where that amount of worms that you have can actually consume the food that you give them quite rapidly. And then you want to give them all kinds of beautiful carbon. That's to develop your fungi. And that could be, as I said, shredded cardboard, could be shredded paper, but also I would be adding leaves from nature and maybe very soft, some sort of a very small, easy to decompose woody material as well. So you don't want to be too harsh with adding huge blocks of, wood, mulch that is sort of hard and made out of hardwood. You want soft materials for the worms to be able to process them, for the microbes to be able to process them, and the worms to feed on them as well. And then over time, as you add 50% this organic matter, that's carbon-based dried leaves and other lovely things like straw and things like that, you just want to build that up with these layers for the worms to feed on. You can also create a Johnson-Su bioreactor, and that's a compost that stays there for a long period of time, once again a year, and you add worms to that as well. And you can look up Dr. David Johnson at the University of New Mexico, and there's a lot of stuff out there how to create this beautiful system. There's a free PDF on the steps on how to do it. Him and his wife have created this and are now really working with farmers to get this incredible biology that it creates and put it back into the farming systems, and they're having excellent results. So we've done one like this at Wood 40 when I ran the mycology research project, and we did a modified version of it. Instead of those chimneys that you could see here that you have to then put in and you've got to take them out to allow aeration, you can do 30 centimeter ring inside, and as long as there's 30 centimeter airflow from any open area, so from the inside of this 30 centimeters in and from the outside of this 30 centimeters in, there will be enough aeration. And we just did mulch. We just sort of mulched or shredded all kinds of organic matter to get some endemic microbes. We then just brought some in as well from a local nursery. And then we filled this bioreactor up. So we've put everything in buckets and put moisture in it. And this is the bought pile as well. And then we lined the bioreactor before filling it with weed matting that allows air to travel through it, but also prevents too much moisture escaping. So you want it to be wet, nice and wet. And setting up a little watering system above it would be ideal. And you basically want it to be wet. You want it to not lose moisture. So you do want to add a spraying system like a sprayer. Dr. David Johnson talks about that and how much to spray and things like that. But basically you want to fill the outside ring. You want to leave the inside ring open, but still protected from moisture loss. And you just fill it up with those buckets with soaked organic matter, and you add it in. But then you wait for it not to be hot, and this never heated up because we never added really much nitrogen. We used mostly leaves and wood chip. So if you're going to add food that's going to heat up, or any manure or anything like that, you want to wait for it to cool down. And once that happens, you can add worms in there as well, and the worms will churn it as well. And over a year's time, this stuff becomes amazing. So of course, you always want to have this analyzed. You can send samples for analysis because I do a whole soil food web analysis, and you assess it and analyze it and measure the soil food web organisms. And this is just a table to show you the exact minimal requirements that we need, these numbers. And this is what we look at, whether we've got those ratios of fungal to bacterial biomass ratio specifically, that we don't have ciliates. If there's a lot of these guys, that means there's anaerobic conditions forming, that we've got more than 100 nematodes, that we have specific number of bacteria and fungi in terms of micrograms per gram. And that's actually numbers of a compost that we created for a trial. I guess it does show you that it's achievable to create quite a lovely fungal to bacterial biomass ratio if you do it correctly. And so basically by analyzing it, I just get final numbers, and we know what's missing. There could be very little fungi, or there could be way too much bacteria, and the conditions might be anaerobic. So you can see in the microscopy analysis the soil food web organisms and how do they relate to soil health and specifically to the plant that you want to produce. And then once you know that the numbers are good and your system's working fine, you can do an extract. So you scrape out some of your compost that you've made, vermicompost, maybe through the worm farm, system I've showed you in the willy bin. When your numbers are right, you can chuck some of this in a little nylon bag, and then you can just massage it with your hand just to dislodge the microbes into a bucket of water. So you're dislodging the microbes for 30 seconds, then you can put that back into the compost or put it under a plant, and this liquid, you can water it into your plant area, or you might want to dunk your plant in this, or you want to just drench the soil before you put the plant in, just to get those microbes into the soil. And you can do this also with the air pump as well and extract the microbes. If you need a larger volume, say you're doing an acreage, then you want to obviously make more. It doesn't matter how much volume of microbes you have in there, because remember, this is an inoculant. You just need them to reproduce. So numbers won't matter as much as whether they can survive and reproduce in that soil that you put them into. So you might have to reapply a few times. It's not a tea, you're not adding any foods or anything. You're just making an extract. You just want to dislodge the microbes into liquid form for easy application. And you always need organic matter in that soil and plants before you apply that, otherwise they won't have food to survive. Then you can just spray them on in whichever way is suitable for you. This is more elaborate application method that you've got to know what you're doing, and you've got to remove your filters from the system, and you can spray it. It's just run on a 12-volt battery operation pump sort of a thing, and you want to mist it out, and all these microbes are going to end up on your crop or your pasture or your soil, whatever you're doing there that you want to apply it to. So basically these are the references. So that first reference I talked about in lesson 1.1, that we took a lot of the research out of that. We talked about the soil organic carbon. You can see there's the reference for that in Western Australian Agricultural Government-based website. And then the State of Knowledge of Biodiversity, that's where you can see all the gorgeous creatures that I've talked about. So there's a PDF of that. And there's also Chaos of Delight by Andy Murray, and wormflow construction and how to feed this wormflow farm that I've written 2 articles about, so you can have a look at that. In our next lesson, we'll talk about bacteria in soil restoration, and this is an exciting topic. There's a lot of great stuff to be learned about the little guys that get nutrients from the rocks and then deliver those to the plants. Specifically, we'll talk about the bacteria that live on the root in the rhizosphere in conjunction with the plant in order to help the plant grow. And so we'll look at specifically plant growth promoting bacteria and the nutrient cycling, how they deliver nitrogen, phosphorus, and potassium, for example, to the plant, and also the extracellular polymeric substances, which are very key gluing and communication substances that allow bacteria to stick to surfaces and produce compounds within that for their communication, and how these worlds of the bacterial life are basically interacting through these substances and what they do for the plant. And the last bit will be rhizophagy, how plant roots actually consume bacteria in order to get their nutrients and then cycle them back into the soil to receive more nutrients. Super exciting. So I look forward to seeing you in the next videos. All right. Thanks so much for listening, and all the best, and see you next time. Bye.